I'd like to discuss with you today the interplay between excitation-contraction coupling and mitochondrial energetics. And you may all know that one main deficit in the failing heart is defects in EC coupling. And let me see how this works. Okay. So, and one important factor of this is that in heart failure, the amplitude of the cytosolic calcium transient is strongly reduced and slowed, and compared to normal calcium transients in normal heart cells. And another problem in heart failure is that there is an energetic deficit, and this has been shown that the phosphocreatinine levels in the failing heart are tremendously reduced. And maybe an even more dynamic parameter, the creatinine kinase flux is reduced in the failing heart compared to normal hearts. And a third problem is that there is increased oxidative stress in the failing heart. And these are tissues, left ventricular tissues from patients with either ischemic or dilated cardiomyopathy compared to non-failing controls. And you see that aconitase activity as a ROS-sensitive enzyme, which is inactivated by ROS, is decreased the activity, and this correlates well with increased lipid peroxidation in these samples. So taken together, these three problems are oftentimes investigated separately from each other, but our focus is to actually see the interrelation between these. And it's well known that oxidative stress can contribute to the defects in EC coupling and also to the energetic deficit. What is less clear, however, is whether defects in EC coupling can actually induce oxidative stress or energy depletion. And this was the center of our interest, since mitochondria are the most important source for ATP, but also for reactive oxygen species, or ROS. And its function is importantly regulated by calcium. So I will talk today about, on the one hand, the physiology of EC coupling and energetics, and then about the topic whether there is a mitochondrial calcium microdomain that explains the interaction between calcium cycling and mitochondria, and whether mitochondrial calcium uptake regulates ROS production, and what changes in heart failure, how changes in EC coupling could possibly result in an energetic deficit and oxidative stress. And while these are mostly published results, I will discuss with you also some unpublished results on the nicotine nucleotide transhydrogenase, a yin and yang of antioxidative capacity in mitochondria. So let's start with the physiology. And during a cardiac action potential, calcium enters the cell that triggers an even greater release of calcium from the sarcoplasmic reticulum. And this calcium activates the myofilaments and induces contraction. And during diastole, calcium is taken back up into the SR by Circa, and from where it can be released at the next speed. And the calcium that came in through the calcium channels leaves the cell via the sodium-calcium exchange. And in normal cells, this amounts about 30% of the overall activator calcium in the cell. And these processes all consume large amounts of ATP, so especially the myofilaments and Circa and sodium-potassium ATPase. And this ATP needs to be replenished by oxidative phosphorylation in mitochondria. So how does that work? Well, substrates fuel into the Krebs cycle, or TCA cycle, whose main product is NADH. And NADH donates electrons to the respiratory chain, and which leads to sequential redox reactions. And this promotes the proton translocation across the inner mitochondrial membrane. And this proton gradient is the driving force for ATP production at the F1, F0 ATPase. Now, when energy demand increases, this is when ADP rises in the heart, for example, when it has to perform more work. This will accelerate electron flux along the respiratory chain and partially oxidize the reduced NADH to NAD+. Now, at the same time, most of the cases you have, when you have an increased workload, when you walk up a flight of stairs, you have sympathetic activation that will increase the cytosolic calcium as well. And this will translate into an increase in mitochondrial calcium. And calcium can stimulate 3 rate-limiting enzymes of the Krebs cycle to increase the turnover rate of the whole TCA cycle. And this accelerates the regeneration of NAD+ to NADH. So these two regulators, ADP and calcium, can very well control respiration from upstream and downstream and maintain both ATP-ADP ratios, but also the redox state of NADH and NAD+ in order to have enough supply for respiration. And now this is now the first point of a debate is now how and how fast does calcium enter the matrix of mitochondria, because the affinity of this calcium uniporter for calcium is rather low in the range of 10 to 20 micromolar, whereas the cytosolic calcium normally is in the range maybe of 1 micromole per liter. So many people have proposed that this is not sufficient to lead to a rapid increase in calcium in the mitochondria. In order to solve this or to elucidate these issues, we developed a technique in which we try to monitor cytosolic and mitochondrial calcium, and in other experiments also the redox state of NADH in the same cell. And we did this by combining the patch clamp technique with fluorescence imaging. And so we loaded cells with the calcium indicator rod 2, which is will, by loading the cells, go primarily to mitochondria because of its positive charge. And however, it always leaves traces in the cytosol. And in order to get rid of these traces in the cytosol, we had to patch clamp the cell and dialyze the cytosol with a pipette solution that does not contain rod 2, but it contained indo as a salt, which works at a different wavelength. So this way we would have two different calcium indicators in two different compartments, namely the mitochondria and the cytosol. And then we could voltage clamp the cells and obtain cytosolic and mitochondrial calcium transients from the same cell. And by doing so, we pace the cells at, in this example, 1 Hertz for here 9 minutes, and then we applied the beta receptor agonist isoproterenol in increasing concentration, and we observed that the amplitude of the calcium transient increases in the cytosol, and the first important result was that during every cytosolic calcium transient, there was also a mitochondrial calcium transient, a rapid one. However, the kinetics of the mitochondrial calcium transient was somewhat different to the cytosol, since the decay kinetics were about 2.5-fold slower than in the cytosol. And the effect of this was that when you increase the amplitude of cytosolic calcium transients in the presence of beta receptor stimulation, you had an accumulation of the diastolic mitochondrial calcium as well. So this was like a pronounced mitochondrial calcium uptake in the face of beta-adrenergic stimulation. Now, the question is, how can such a rapid mitochondrial calcium transient occur, given that the affinity of the calcium uniporter is so low? Well, one way to explain this would be if you have a closer look at the texture of a cell, you will realize that mitochondria are here very closely associated with the sarcoplasmic reticulum. It's literally enwrapped, and the distance between the SR and mitochondria is less than a micrometer, in the nanometer range. And it is also known that in the nanometer distance from the ryanodine receptor, there are calcium concentrations occurring for a very short time that are far beyond 1 micromolar, but rather between 10 and 100 micromole per liter. So this close association would be a microdomain in which the very high calcium and at a very coordinative calcium release would be sufficient to overcome the low affinity of the calcium uniporter. And to test this further, we compared two conditions, namely one, a rapid SR calcium release, either by triggering it via the calcium channel or by applying caffeine. And we compared this condition to a condition in which calcium, where the SR was depleted with thapsigargin, so there is no calcium in the SR, and then by using the reverse mode of the sodium-calcium exchanger, we slowly brought in calcium from the cellular membrane, so a different location and a different velocity. And by comparing this, so this is a protocol, this is a voltage step to +10, which will induce an L-type calcium current. And this induces a rapid cytosolic and mitochondrial calcium transient, and this is the protocol where we step to plus 80 millivolts in the presence of thepsigargan. This induces an NCX current and a slow and gradual increase in cytosolic calcium and also in mitochondrial calcium. But you also realize that in order to achieve the similar increase in mitochondrial calcium, it took about twice as much cytosolic calcium when this was brought in slowly via the sodium calcium exchanger. And if you now plot the increase in mitochondrial over the increase in cytosolic calcium in this very same cell, then you obtain slopes, and if you calculate these slopes, this can be used as an estimate for the efficiency of mitochondrial calcium uptake. And this efficiency was highest when there was a coordinated SR calcium release and was more than 50% decrease when calcium came in slowly via the NCX. And so this would be in favor of such a microdomain between the SR and mitochondria where vicinity or location is important, and also the velocity of a calcium flux. Now, to test whether this has any bioenergetic consequences, we performed an experiment in which we again compared the SR calcium release, this time by caffeine, with a reverse mode NCX current. And to control for equal cytosolic calcium, we calculated this integral. And I tell you that this is the actually indicating the amount of cytosolic calcium in this case. And we also double-checked by doing a set of experiments where we actually monitored cytosolic calcium with Indo. So under these conditions, similar increases in calcium occurred in the cytosol. However, when looking at the redox state of NADH and FAD, which in this case are a monitor of mitochondrial matrix calcium activating the Krebs cycle, we observed that only after caffeine, but not after the NCX pulse, we had a reduction of oxidized NAD plus to NADH, which you see here, and for FAD this goes the opposite way because a reduction is a decrease in fluorescence, and you can make the ratio out of both of these, and both indicates that there is a calcium sensitive activation of the Krebs cycle that takes place after SR calcium release, but not after a slow and diffusional influx of calcium over the cell membrane. Now, in a project that is now going on in cooperation with Gerald Dawn, we're trying to look for the molecular determinants of such a microdomain, and it has been observed in fibroblasts that mitofusin 2, MFN2, is a linker protein, in that case between the ER, the endoplasmic reticulum, and mitochondria, like a physical linker. So Gerald Dawn actually developed a knockout mouse that has a cardiomyocyte-specific knockout of mitofusin, and we tested these mice by performing experiments in which we measured the sarcomere length in a protocol where we pace the cell from 0.5 to 5 Hertz and then step back to 0.5 Hertz. And in the presence of the beta receptor agonist isoproterenol. And we observed no difference between the mitofusin knockouts in terms of increase in sarcomere length, fractional shortening, time to peak, or RT50. So the cardiac myocytes performed the same amount of work. However, this is still preliminary data because this is just out of 4 and 4 animals. But these are the patch clamp data that indicate that despite similar cytosolic calcium transients in these mice, we had, especially in the early phase of isoproterenol wash-in and 5 Hertz stimulation, a trend towards decreased mitochondrial calcium uptake. But as I said, these data are still preliminary. But when looking at the bioenergetic response in terms of NADH and FAD, we observed that in the mitofusin knockouts, there was an initial oxidation of this redox state, which could be explained by isoproterenol inducing a positive inotropic effect that increases ADP, which accelerates respiration and oxidizes NADH. And since mitochondrial calcium uptake under these conditions was not as good as in the wild type, the compensation that calcium induces by accelerating Krebs cycle is less pronounced. So the net result would be an oxidation of the redox state of both NADH and FAD, and which could lead to at least transient energetic deficit. And well, in the long run, these animals develop cardiomyopathy that seems to have a mitochondrial cardiomyopathy, but Gerald Dorn has more data on that. And but this would just be one piece in the puzzle where a microdomain could be important to control mitochondrial calcium uptake. And in fact, this microdomain could be related to the physical linker between the SR and mitochondria, which could be obtained by mitofusin 2. So the second topic I'd like to touch on is how does mitochondrial calcium uptake regulate ROS production? Well, to understand this, let's have a look at ROS production in myocytes in the first place. So at the respiratory chain, superoxide is generated at complexes 1 and 3, and superoxide is dismutated to H2O2, and in order to eliminate H2O2, it takes a whole array of enzymes that in the end all require reduced NADPH. This is the glutathione and the thioredoxine systems here. Now, it is interesting to note that the regeneration of NADPH depends, as far as we know today, in mitochondria on 3 enzymes, which is the isocitrate dehydrogenase, the NADP-dependent form of it, malic enzyme, and the transhydrogenase, which I will talk especially in the last part of my talk also. And all these enzymes derive their substrates from the Krebs cycle, which is NADH, malate, and isocitrate. So we speculated that mitochondrial calcium uptake may be important not only to match energy supply and demand, but also to keep the redox state in a reduced state in order to prevent the formation of toxic H2O2. Now, to test this, we again applied the technique in which we, together with the patch clamp technique, either monitored cytosolic and mitochondrial calcium in the same cell, or in a second protocol, we monitored the redox state of NADH together with the H2O2 sensitive indicator DCF. And we performed a protocol in which we paced the cells at 3 Hz and then washed in isoproterenol at 2 different concentrations. And isoproterenol will increase the amplitude of the calcium current in this case. It will increase the amplitude of the cytosolic calcium transient, and it will also lead to an accumulation of mitochondrial calcium. This is only the diastolic trace here. There is also beat to beat, but this is the diastolic accumulation. And then what is interesting to see is that at the onset of work with isoproterenol, there was an initial oxidation of NADPH, which we think, and there is data by Brunnes and Burrs on that too, that initially it is this oxidation of NADH is due to ADP inducing an acceleration of respiration. And after this initial oxidation of NADH down to 34% after 1 minute, there was a continuous recovery, and this recovery was temporarily closely related to the increase in mitochondrial calcium. So this is also within the general idea that mitochondrial calcium accelerates NADPH regeneration via the Krebs cycle. Now, when looking at ROS production during this time where you have this temporal oxidation of NADH, NADPH, by the way, this is the fluorescence of NADH and NADPH cannot be differentiated in one cell, so it's the overall NADH and NADPH signal. So during this oxidation of NADPH, we saw a slight increase in the ROS production. You may not see this here, but if you have a look at this graph here, this is the H2O2 formation rate, so the change of fluorescent within 1 minute. And at minute 5 is the minute before isoproterenol hits. At this minute, we have a reduced NADPH redox state and low H2O2 formation. Now, 1 minute later is when you have this transient oxidation down here. This is when NADPH oxidizes. In this minute, H2O2 formation, the rate of it is increased by about 5 to 6 fold. And at minutes 7, 8, and 9, with continuous mitochondrial calcium accumulation and recovery of NADPH redox state, this H2O2 net formation is reduced back to normal again, and in the following, at minutes 12 to 17, maintains at a low level, which is comparable to the level before isopropernol. So we think that this mitochondrial calcium uptake by stimulating the Krebs cycle prevents an overshoot of H2O2 formation by keeping the redox environment in the mitochondria more reduced, which will enable antioxidative enzymes to eliminate H2O2. Now, as a proof of concept, we added an inhibitor of the mitochondrial calcium uniporter, Ru360, in the pipette solution, and this inhibits mitochondrial calcium uptake, and it blunts the recovery of NADPH and provokes an increase in H2O2 production in the mitochondria, indicating that calcium uptake indeed is important to control H2O2 production. Now, what changes in heart failure? Well, as I said before, the calcium, or I didn't say it, the calcium load of the sarcoplasmic reticulum is strongly reduced. On the other hand, the sodium concentration in the cytosol is elevated, and this, on the one hand, leads to a reversal of the sodium-calcium exchanger, which can then bring in calcium instead of exporting it during the action potential. And that's a lot of traces here, but it's from a study of Antonis Amundas. But if you look just at the magenta line, this is a normal heart at a normal sodium, and this is a failing heart cell at a higher sodium that these hearts have in a patch clamp experiment. And the line here, everything that is below this line is calcium extrusion by the sodium calcium exchanger, and everything above that line is calcium influx via the NCX. So you see that in a failing heart cell, for most of the action potential, there is calcium influx via the sodium calcium exchanger. Now, it has been assumed that this is a compensatory mechanism that may compensate for decreased SR calcium load, but from an energetic point of view, it may be more adverse, because I've shown you this data already. A slow NCX-driven calcium influx is less effective in mitochondrial calcium uptake. On the other hand, this increase in sodium itself may also play a role, because the calcium elimination from mitochondria is driven by a sodium-calcium exchanger as well. And since sodium when this is elevated, it drives calcium out of the mitochondria. And we observed previously that this leads to an oxidation of NADH. And this is this experiment where you have an increase in sodium concentration in the filled dots, and the normal sodium in the open dots. And you see that despite similar cytosolic calcium, there was less mitochondrial calcium and a more oxidized redox state of NADH. So we speculated that this, according to what we've observed before, may provoke H2O2 formation, and in fact, by increasing the sodium concentration in the cytosol of a healthy myocyte, there was an increased formation of H2O2, especially at the onset of work here, that may translate into oxidative stress. So increased sodium could induce oxidative stress. Now, to test this in a guinea pig heart failure model induced by ascending aortic banding, they have a hypertrophy and decreased left ventricular systolic function and an elevated sodium concentration in the cytosol. And in these cells, H2O2 formation in a protocol similar to what you've seen before was strongly increased compared to non-failing myocytes. Now, if you block the export of calcium via the sodium-calcium exchanger with the CGP compound, then this increase in ROS production could be reduced again. And as a second proof of concept, when patch clamping these cells and dialyzing the cytosol with a sodium concentration of 5 millimolar instead of 15, this would also reduce mitochondrial ROS production in these circumstances. So that we propose a concept in which the increase in sodium in heart failure may lead to less mitochondrial calcium uptake and to more mitochondrial calcium release. And this reduces the steady-state mitochondrial calcium, which leads to less stimulation of the Krebs cycle and to a more oxidized NADH and NADPH redox state. And this can give rise to H2O2 formation, which in turn could initiate a vicious cycle. For example, since CAM kinase is known to be redox sensitive and to promote an increase in sodium by activating the late sodium current. So this could be potential drug targets to that could also maybe be helpful in heart failure to prevent this vicious cycle of energetic deficit and EC coupling dysfunction. So I'd like to make a little cut here, because now we shift a little bit away from the calcium, but you still will need this, what we discussed now, to understand the issue of the transhydrogenase, because as I've shown to you, the transhydrogenase seems to be an important enzyme that actually mediates this the coupling of the antioxidative capacity to the Krebs cycle activity. Now, it was our initial idea to maybe target this protein by a drug. There is a drug that inhibits it, but it inhibits also the ATPase, so it's not very useful. So we thought, well, maybe one could make a knockout animal or so to knock it out genetically. So I went to the literature and I looked, and this was around 2008, and I realized that there actually was a mouse that is deficient of the transhydrogenase, and it's not just any mouse, but it's the C57 black 6 mouse from Jackson Laboratories, which is the most commonly used mouse for any models used in not only in cardiology but in all terms of research. At least that's what you can read on the Jackson homepage. And well, it's been known that these mice have an impaired glucose tolerance, and with the development of genetics, people used a quantitative trait loci analysis to actually, in which you crossbreed these mice with other mice, and then you get a totally random F2 generation where you have all kinds of lines that you can then look at your phenotype in, and then you can follow back your defect that this mouse has in the first place and see on which chromosome the problem is sitting. I'm not a geneticist, so there are others that can explain that better. But in the end, you end up with a chromosome or even a chromosome locus where you know the defect is sitting. And they came up with chromosome 13, and actually with the NNT that would account for this defect. And when taking a closer look, they found a mutation in the NNT gene that leads to the loss of exon 7 through 11 and a dysfunctional protein. And in a cell metabolism paper from 2006, they could show that in a cell system, when you knock out the NNT, you get increased oxidative stress and uncoupling of ATP production and less efficient insulin secretion. So that means when glucose is coming after having lunch, the insulin secretion is impaired in these animals, and they are more likely to develop diabetes. And in the same year, a second study observed that when you knock out the SOD, which eliminates superoxide, or dismutase superoxide, is knocked out in black 6 mice, these mice die even before, at least either before birth or shortly after 2 days, whereas all the other backgrounds, they survived for at least 14 days. So, and they also came up with the NNT. So the thinking is, up to this point, that the NNT is important as an antioxidative enzyme to prevent oxidative stress. So we said, that's great, we can use it and prove our concept that the NNT is important to protect from oxidative stress. So we checked whether it's really the case that exon 7 through 11 are lacking. You do this by PCR analysis, and you see that we compared the black 6J mouse to a black 6 mouse that was diverted from that J strain in the 70s. And this is actually the black 6 mouse we get in Germany when you buy them at Charles River. And they have a full-length NNT, so they don't have the defect, but the J mouse, they have this defect. So we use this one as a control mouse because it's very similar to the J mouse, and except for this difference. And we confirmed by enzymatic activity that indeed the NNT activity is gone in the J mouse, but it's still there in the N mouse. And to see whether there is a compensatory upregulation of the other enzymes that can regenerate NADPH, we checked for isocitrate dehydrogenase and for malic enzyme. And we observed that there was no compensatory upregulation of these enzymes in the J mouse. But when you look at the overall maximum rates, we observed that actually the isocitrate dehydrogenase was quite effective in performing this reaction. So when combining the Vmax values of all these three enzymes, we came up with a very crude calculation of the contribution of these enzymes to NADPH regeneration, which would amount dehydrogenase. So the NAT would contribute about 28% to this overall NADPH regenerating capacity. So it's not the major player, but maybe an important player. So then next step was that we isolated the mitochondria from these animals, and we performed three measurements, namely respiration with a Clark electrode. So that means how well is the electron transport chain functioning. Then we measured the superoxide generation with a spin trap technique, and the H2O2 formation in the extra mitochondrial compartment with the amplex red assay. And especially the elimination of H2O2 is dependent on NADPH, where our NNT is actually coming into play. So when looking at respiration, well, the way you do these experiments, and we have experts in the audience that do that every day, but for those who aren't, what you do is you add the buffer into a chamber where you have a seal, and you measure the oxygen in there. And then you add the mitochondria, and when you only add mitochondria without anything else, not much is happening with oxygen because there is no need to respire. And then you add substrate, then you get some respiration, which is considered to be due to uncoupled respiration. But the real thing is if you add ADP, which I've told you before is accelerating respiration very efficiently. So you see when you add ADP, you get a steep increase in oxygen or a decrease in oxygen tension, which means oxygen consumption. So this is respiration. And then at the end, you calibrate the system by adding cytochrome C. Well, to make a long story short, oxygen consumption with the Clark electrode was not different in state 3, and the respiratory control ratio was also not different between these two strains, meaning that the respiratory chain is probably not defective. And when looking at a superoxide generation with the spin trap technique, we observed similar amounts of superoxide generated under these different conditions. Now in this context, it's important to explain that the general concept of ROS production in mitochondria is that most superoxide in the first place is generated when the redox chain is highly reduced. This is a situation when you don't have ADP, but a resting cell, and you have substrate present that gives you a high NADH, and all the electrons are sitting here in the respiratory chain, but there is no need to respire because no ADP is present. So what do the electrons do sitting around here? They prematurely produce superoxide. And this superoxide generation can be reduced when you oxidize the respiratory chain, which means you add ADP, which will make the electrons flow the normal way to the cytochrome oxidase to go to oxygen to form water and not superoxide. And this will reduce superoxide. And this is what you see here when you step from state 2, which is in the absence of ADP, to state 3, which is in the presence of ADP. So this, adding ADP, reduces superoxide formation in the first place. Now, when looking at H2O2 formation, the picture is a little bit different. First of all, when you look at the normal mitochondria, which have a transhydrogenase, here, when you step from the absence to the presence of ADP, there was no decrease of H2O2 formation, like with the decrease of superoxide, but it maintained H2O2. Now, how can this be explained? Our idea is that when you uncouple or when you accelerate respiration with ADP, you reduce superoxide, but at the same time you oxidize NADH and presumably also NADPH, and this will reduce the antioxidative capacity and make H2O2 formation more likely. So you increase H2O2, but you lower superoxide by adding ADP. And this could explain why the overall H2O2, which is measured outside the mitochondria, is actually staying the same. It's at this point a hypothetical explanation, but we have some evidence that may be in support of this. Now, the important observation is, although H2O2 formation was not significantly different between these two strains in the absence of ADP, in the presence of ADP, H2O2 formation now decreased in the J mouse. And the way we would explain this is when you accelerate NADH coupled respiration, you reduce superoxide. This is within the concept that is already established. But when this enzyme is lacking, it seems that the NADPH is conserved. And this means that if it is there, and you have NADH, you have an acceleration of NADH coupled respiration, what happens is that the transhydrogenase reaction actually reverses. So you see that now, when NADH is consumed to NAD+, you have a substrate for NAD+ and NADPH, and you have less product. This will favor the reaction of the direction of the reaction to reverse and consume NADPH in order to support NADH coupled respiration. But this concept has not been out there yet, so this would be a new idea how this could work. Now, and this, so in our J mouse where this is lacking, you reduce superoxide, but you maintain NADPH because it is not consumed by the NNT, and this would also reduce the overall H2O2 formation then. So this would be our explanation why in this experiment H2O2 production actually was decreased just as the superoxide was decreased from state 2 to state 3 because the NADPH pool was conserved. Now, to prove this idea, or to get more evidence for this idea, we performed an experiment in which we used isolated cardiac myocytes. And another way of accelerating respiration instead of ADP is, which is difficult to do in a cardiac myocyte, is to add FCCP. And FCCP uncouples the proton gradient just as this here is doing. And this will also lead to an oxidation of NADH. And by doing so here we add FCCP, and then we measure NADH, and we also measure FAD. This is going the other direction. You remember this from the experiments before. And we do this in the N mouse with the NNT, and in the J mouse without the NNT. So we add first FCCP, and then cyanide, and cyanide will do the opposite. With cyanide, with FCCP, you oxidize the redox state of NADPH and you oxidize FAD. And then you wash out FCCP and we add cyanide. And this is doing the opposite. So the uncoupling is gone, and with cyanide you block respiration, and this will lead to maximal reduction of NADH and NADPH. So with that, you fully reduce the redox state in the cells. You see a full reduction of NADH and of FAD. And then we added H2O2. And H2O2 will also consume all the NADPH and NADH from the other side, not from here, but from here. And you see that the overall oxidation of this redox state is the same with H2O2 as with FCCP in the mice that have the NNT. Now the important difference is between the mice with and without the transhydrogenase is that when you add FCCP, we did not achieve the full oxidation of the overall NADH-NADPH pool, presumably because the NADPH was conserved because of the lack of the NNT. Whereas the H2O2-induced oxidation was similar as in the N mouse, and we monitored for the full oxidation by monitoring FAD, which is independent of NADPH, and which is a good indicator of the respiratory chain. So at equal oxidation of the respiratory chain, we had a higher oxidation after H2O2 than after FCCP under these conditions. So when putting this into a simple bar graph, it means that the FCCP sensitive redox state of combined NADH and NADPH is smaller in the mice that lack the NNT, whereas the overall H2O2 oxidizable redox state is the same as in the N mouse, and that is the same under both conditions in the N mouse as well. So this means that about 23% of this overall redox state is due to NADPH, and it would also prove that the when this enzyme is lacking, no NADPH oxidation takes place in the face of an acceleration of NADH-coupled respiration. This may be the most difficult part of the talk. From now on it gets easier, but this would be the concept. So what is a situation in vivo where you have an increase, an isolated increase in workload? Well, it's maybe difficult to get something like that in real nature, but experimentally it's been shown already by Brunnes and Burrs that when you increase the sarcomere length of a trabecula, you actually this leads to an oxidation of NADH and of NADH. Whereas if you increase calcium or the frequency, this is what we observed before, this undershoot and recovery behavior, you get an oxidation and a subsequent recovery because calcium comes into mitochondria and accelerates respiration. And here it's the frequency-dependent accumulation of calcium in the mitochondria that regenerates the redox state. Whereas here, when you only increase the workload without changing calcium, you get only the oxidation without the compensatory increase in the redox state driven by calcium. So in vivo, what you can do is you can do an aortic banding in an animal to increase the workload without primarily affecting calcium homeostasis necessarily. And we did this. So we did aortic banding in the mice, in the N and the J mice, and we followed them for 6 weeks. And after 6 weeks, we analyzed them. So we had similar increases in heart rate by tibia length ratio. And these are 44 and 47 animals and 20 shams. And there was a slight difference, but it was not statistical. It was not different statistically. And, but when looking at the pulmonary fluid as an index of pulmonary congestion, we realized that 21% of these animals were decompensated in the mice that had the NNT and 6% that did not have the NNT. And I don't know if you see this, but when looking at fibrosis, serious red staining, there was a much higher degree of fibrosis in the mice with the NNT, the black 6N strain, compared to those that did not have the NNT, while myocyte hypertrophy was of similar increase. An important regulator of fibrosis is CTGF. And CTGF was about 40-fold activated in the N mice, but only about 6-fold in the J mice. And even when this is a data set with some preliminary data of this, but even in those animals that had a high increase in heart rate to body weight ratio of this group had a clearly reduced activation of CTGF. And CTGF is known to be activated by H2O2, its expression. So we looked at H2O2 in these hearts, and we did this by 8-hydroxyl D-guanosine staining. And this is a staining that will turn red when there is a reaction with H2O2. So this we observed that in the TAC. These are actually all from animals with after aortic banding, and these are 2 different examples from a J mouse, and these are 2 examples from an N mouse, and you see clearly, especially in this sample, that there was a much higher likelihood of red staining in these tissues than in the J mice, and when quantifying this, the percentage of HODG positive cardiomyocytes in the nuclei was tremendously increased in the N mice, but not at all in the J mouse. So this would be an indicator that what we've observed on the isolated mitochondria level, where ADP, that represents work, induced about 2-fold less increase in H2O2 formation, could translate into less pronounced oxidative stress after 6 weeks of aortic banding. When looking at, you remember pulmonary congestion was high. This is normally a sign for increased left ventricular filling pressures. And another biochemical marker for this is ANP, which is elevated with increased left ventricular filling pressures. And this was significantly increased in the N mice after aortic banding, about 20-fold. Whereas it's hard to believe, but it's true that it was not significantly increased, but you see it was at least less increased in the J mice. And when looking at left ventricular ejection fraction, which was determined by pressure volume loops in a working heart system, this was reduced after 6 weeks in the N mice after aortic banding, but preserved in the J mice. So taken together, and I think with this I'd like to end, we learned from this that, oh no, there is still a little bit, sorry, I forgot. So a selective increase of work might actually reverse the NNT reaction, and in this context, the NNT may actually promote oxidative stress by consuming the protective NADPH in order to support NADH-coupled respiration. Now, as I said, most of the times when you have a physiological workload, you have beta-adrenergic stimulation. Under these conditions, you have an increase in calcium in the cytosol, in the mitochondria, and an acceleration of the Krebs cycle. So the question is, what happens if you have a more physiological increase in workload? And here, well, we performed experiments on our setup with isolated cardiac myocytes, and you've seen some data from that setup already. The key is to have a camera where you can monitor the sarcomere length and 3 photomultiplier tubes with which you can monitor the different wavelengths of NADH, FAD, and all the other dyes. And well, this is the protocol, and I've shown you this before. This is again this protocol where we, maybe I show it here once more. So it's in this case a 0.5 and 5 Hertz stimulation protocol in the presence of isoproterenol in order to induce a rather physiological increase in workload, because you have an increase in rate and amplitude of cytosolic calcium transients. And then we can calibrate the system with FCCP, cyanide, and H2O2 afterwards. In this example, we combined NADH with DCF. And when you do this in these myocytes with NNT and without NNT, you get similar increases by isoproterenol of the sarcomere shortening, and also mitochondrial calcium uptake was similar in both strains. And also the overall NADPH, NADH redox state and FAD was not different between both strains. Also, if you make the ratio out of both, which is this graph, you see that this is the most sensitive indicator we have for the redox state here in the mitochondria. It was not different between both strains. However, as I said, it does not report us how much from this signal is NADPH and how much is NADH. In order to get that, we need to look at something that is only NADPH dependent, namely the elimination of H2O2. And now, under these conditions, despite a similar increase in superoxide generation, which is by mitosocs, the H2O2 formation reported by DCF was increasing in the mice that lack the NNT. So now it's the other way around, because now we have a condition in which we push the activity of the Krebs cycle to reduce NADH, and in this situation the whole reaction goes in the forward direction. And when it's lacking, then something is missing that supports NADPH dependent elimination of H2O2. And to look for this in vivo, we applied isoproterenol in vivo for, oh actually this is wrong, this is for 2 weeks. And after, it's not there, and after 2 weeks, we had similar increases in heart rate by tibia length, although there was a trend towards more increase in the J mice that lacked the NNT. So at least it was not in the same direction as after aortic banding, where you had a less severe cardiac remodeling. In this case, the remodeling is similar, if not a little bit more in the J mouse. So taken together, it means that the NNT has different functions. So on the one hand, it's important to regenerate NADPH in conditions where especially Krebs cycle activity is high, and maybe also conditions where energy consumption is not so high. If we go back to this diabetes problem, this is a situation where you have the pancreatic islet cells in the absence of glucose, while the redox state is more oxidized, and then glucose is coming in, all the substrates will accelerate Krebs cycle activity, and there is not so much ATP consumption because these cells don't work so much. So this is a situation where this direction is strongly favored. So in this condition, when this is lacking, then you have oxidative stress. And we also observed that in the pancreatic islet cells, this is difficult to understand, so the bottom line is the NNT in these cells, other than in the cardiac myocytes, is playing the dominant role, because in the presence of ATP, the isocyte dehydrogenase is completely suppressed. This is a physiological feedback regulation. So this means the NNT is the only enzyme that can account for regeneration of NADPH. So when this is now lacking, this cell under these conditions will have increased oxidative stress, whereas in what we've observed in the heart, where you have high energy demand, and without compensatory calcium-induced acceleration of Krebs cycle activity, this can all switch the other way around and then consume NADPH and lead to oxidative stress. And with this, I'd like to thank you for listening patiently, and I'd also like to thank my group. Most of this has been work in the first part by Michael Kohlhaas. And the NNT project has been done especially by Alexander Nickel, Albrecht von Hardenberg, and Michel Goulens, our technician, who is doing an excellent job on the isolations. And the mitofusin project has been done by Maximizer. And I'd also like to thank the cooperators, Gerald Dawn and Brian O'Rourke in Hopkins and Washington University. Thank you very much for your attention. I haven't looked too deep into this, but I read there was a recent cell metabolism paper on aging, and also on the role of isocyte dehydrogenase. But in this paper, it said that the black 6J mouse is the most common aging model. So this would be in favor of the idea of NNT in most organs as an antioxidative enzyme, because many people associate aging with increased oxidative stress. But I don't know if this is the only reason why they use that mouse, but it is interesting to note that this particular mouse is used commonly for aging studies. But I'm not an expert on that.
Could be more or less neutral.
The same, right. In this respect, I think it is important to relate the increase in cytosolic calcium to the increase in mitochondrial calcium, because in the situation that you talked about and that we also tested experimentally, the amount of calcium that goes into mitochondria at the same cytosolic calcium is less when this is sodium driven. So if you increase sodium, you may get a compensatory increase in calcium, but calcium also means work, because also for the time of the relaxation. So we know that the relaxation in the failing heart is slowed. And even a prolonged relaxation is a high burden of work. And that's been shown by Gerd Hasenfuss a couple of years ago, that even the resting heart consumes energy because of the elevated diastolic calcium in heart failure. And this energetic burden is not to the same degree matched by mitochondrial calcium activation. So it's a relative issue. You are right that on the one hand, the more calcium should also push up the calcium in the mitochondria a little bit further, but the relation is shifted. There is less mitochondrial calcium for the same amount of cytosolic calcium when the sodium is higher. And I think this plays a role. Although there is maybe the same, even if you create the same mitochondrial calcium influx, like in our experiment, this is at the cost of higher cytosolic calcium, which induces more work and more ADP-induced oxidation of NADH. And then you get the oxidation of the overall. So it's always a question of balances.